students in the Sanjeevanee Mahavidyalaya's YouTube channel. Today we are going to study the genetic material. As you know that DNA and RNA are the genetic materials, but under that we'll see the point evidences for DNA as a genetic material. With the help of three experiments, Griffith Experiment, Avery McLeod and McCarthy experiment, and Hershey and Chase experiment, we can say that DNA as a genetic materials. Let us see the first introductory part. First time the scientist George Mendel, he carried out experiment, or George Mendel's, he discovered some principles and mechanism of genetic inheritance, and he said that the genetic material transferred from one generation to another generation in the living things, but the chemical nature of the hereditary materials remained unknown that times. During that times, number of scientists, they said that proteins are genetic materials, because the chemical analysis had shown that proteins are more varied than the DNA. That's why they thought that proteins may be genetic materials. And that's why later on the discovery of DNA as a hereditary material came as a surprise to scientists. And with the help of experiment on bacteria and bacteriophages. Bacteriophages means viruses. With the help of these two microorganisms, number of scientists proved that how the DNA is a genetic materials. First time in 1918, a Spanish flu pandemic. It means that Spanish flu pandemic, it is the disease that time spreaded in the world. And in England, the scientist, microbiologist Frederick Griffith was studying the two strains of Streptococcus pneumonia. Strains means one variety of two different kinds of microorganisms that varied in both their appearance. One Streptococcus pneumonia is a pathogenic, and another Streptococcus pneumonia is a non-pathogenic. And that time the scientist Frederick Griffith, he carried out an experiment for curing a Spanish flu pandemic disease. He said that the highly virulent S strain had a smooth capsule. Means smooth colonies on media. It is a virulent, it is a pathogenic. And there is the another strain, R strain. It is a non-virulent, it is a non-capsulated, and rough colonies on media. Means two different strains of Streptococcus pneumonia he selected, or he studied during his experiment. Frederick Griffith was a British bacteriologist. In 1928, he performed experiment which is now known as Griffith Experiment. And with the help of this Griffith Experiment, he proved that DNA as a genetic materials, and in his experiment he selected which microorganism? Streptococcus pneumonia, which is a causative agent of pneumonia. Nowadays we know that due to the coronavirus, due to the COVID-19 disease, the first symptoms, it is the pneumonatic symptoms. Pneumonia, it means that if the people are suffered from pneumonia, maybe 90% maybe we can say that there is some relations with coronavirus. It's like that. Coronavirus it is advanced stage than the pneumonia. It is the next stage than the pneumonia. And there are two strains he selected in his experiment. This is the requirement of Griffith experiment. Basically, two requirements. One, two strains of Streptococci pneumonia, and second, experimental animals, that is the mice. First is the strain Streptococci pneumonia, and in the Streptococci pneumonia there are two strains, live smooth S strain, live raw R strains. Live smooth strains, live rough R strains. S means pathogenic, virulent encapsulated strains. R means it is a rough colonies, it is non-capsulated, it is a non-pathogenic strains. And apart from that, he also used heat killed smooth S strain and heat killed R strains. So how these strains are he used in his experiments, that we'll see in his experiment. This is the some idea about the experiment. This is the how he carried out experiment on mice. You just see in the first, he selected the smooth strain. Smooth means that is the Streptococci pneumonia S strain. S means pathogenic, S means virulent, S means encapsulated. He injected S strain to the living mice, live mice. What happens when once injected the S strain into the mice, after a period of times, what happens? Mouse contracts pneumonia. Means the mouse or mice, it becomes pneumonatic, and that's why after a period of times, the healthy mouse, it becomes dead due to the S strain. It is pathogenic. After that, the Griffith scientist, he used another strain, another microorganism, that is the Streptococci pneumonia R strain. R means it is a non-pathogenic, it is a non-capsulated, it is non-virulated. He again used that R strain to the another mouse. That R strain injected into the mouse with the help of injection. What happens? Mouse remains healthy. It means that there is no side effect of that R strain on mouse. Mouse remains healthy after a period of times. It means that there is no side effect of R strain. Why? Because it is a non-virulent strain. After that, in the third experiment, what happens? It means in the third step, the scientist Griffith used a heat killed S cells. Heat killed S means smooth cells. Smooth means pathogenic. Means it is a virulent. And when he used that strain to the mouse, what happens? The mouse remains healthy. Due to the heat killed process, what happens? Whatever the virulent capacity of the pneumonia, whatever the pathogenic capacity of the pneumonia, it destroyed, or due to which what happens, the bacteria are killed due to the heat kill process. That's why mouse remains healthy. And in the last step, what happens? He used mixed strain. Mixed strain means living R cells plus heat killed S cells. He mixed these two strains for experimental animals. It means he used a mixed strain. He injected this mixed strain to the healthy mouse. What happens? Mouse contracts pneumonia. It means that he suffered from pneumonia, and after a period of times, what happens? The mice or mouse, it killed, it died due to the injection. Why? During the mixing of R strain and heat killed cells, during what happens? Whatever the virulence activity of the strain, it transferred into the living R cells. And what happens? What happens? In the injection, there is the pathogenic activity, and once they injected into the mouse it becomes dead due to the mixing strains. And with the help of this experiment, the scientist Griffith, he proved that there is the genetic material DNA is present in the streptococci pneumonia in the S cell as well as R cells. And with the help of these four steps, he proved that yes, DNA is a genetic material which is responsible for transformations of what? Transformation of genetic materials, that is pathogenic as well as non-pathogenic, due to which what happens? We can observe such type of things, such type of result. In the first result, there is a S colony is isolated in the on the media. In the second, R colony is isolated. In the third, no colonies. Why? Because due to the heat kill process. And in the last step, you just see the plate on which there is the mixing, there is the growth of colonies of mixed microorganisms. This is all about the evidences. DNA is a genetic material. The first time the scientist Griffith, he proved that in his experiment DNA is a genetic material. Thank you.